All right, guys, so you may recall we were working on this prokaryotic envelope that exists on the outside of prokaryotes. It includes possibly a capsule or a slime layer, not always, but then we move down in a little further and we oftentimes run into a cell wall. Now we talked about how the cell wall could be thick for gram-positive or thin with gram-negative. These are made of peptidoglycan, so partly protein, partly sugar. But now we're kind of continuing our way in here, and we have to talk about the plasma membrane. And here on this image, this is actually showing you two membranes in yellow. So the plasma membrane, typically we're talking about this inner membrane that is around the cell. This outer membrane is only associated with gram-negative organisms. So we know that based on this picture that this has to be a gram-negative organism with a thin peptidoglycan layer, right? This is this is an electron microscope, transmission electron microscope image showing you where that peptidoglycan layer happens to be. But now we're focusing on these plasma membranes. So here we see in the bottom left a typical plasma membrane. It's not that much different than what you see in a eukaryote. It has these phospholipid bilayers, right? So we have these partly lipid molecules. And so I'll just draw off to the side here. We have these hydrophilic parts of a phospholipid molecule, and then we have the fatty tails, the fatty acid tails that face one another. They would be hydrophobic, right? These are things you're looking at in your chemistry assignment. So the hydrophobic would be towards one another. Makes sense. They don't want to be facing in or out of the cell because it's hydrophilic environment out there, right? There's water on the outside of the cell and there's water on the inside of the cell. So this could very well be around a eukaryotic cell. But if we consider the membrane, whether it's a plasma membrane on the inside of the gram-negative organism we just looked at, or whether it's on the outside, there are a lot of similarities. So here we see how it is that with the outer membrane, we'd have a similar situation for a gram-negative organism, right? And then here we have that peptidoglycan layer and a periplasmic space. And then we have, again, a similar situation, these phospholipid bilayers making up membranes, right? Now, what can we say about a plasma membrane, or the outer membrane for that matter? We can say that they're made mostly of phospholipids that we just talked about. There are proteins in here, and this is a fluid mosaic model, right? Which basically means that these proteins, some of which go clear through the membrane, allowing things to move in and out, some of which are just on one side or the other, but they're like ships in the sea and they're able to move. Some of them are more static, but because many of them move, we say fluid mosaic model, like ships in a sea. They can move around. And like a membrane is with a eukaryotic cell, it can be selective about what it allows to move through that boundary, right? So this is a boundary to the inside of the cell, and it also keeps certain things inside that it doesn't want it to get out. Now, in our discussion, though, some significance about when we talk about these membranes lends itself to our ability to control growth and contamination. So if we just consider how it is that the membrane, inner or outer for that matter, and in the case of a gram-positive organism, there will only be one, right, on the inside of the thick cell wall. What do alcohols do, right? So when we apply alcohol or an alcohol swab to the skin, what do we mean to be doing there? We're trying to sanitize the area, right? So we're trying to kill a bunch of bacteria that happen to be in that region. And what are these alcohols doing? Well, they have a kind of a amphipathic kind of characteristic. In other words, they have a little bit of a nonpolar, uncharged characteristic and a little bit of a polar characteristic, just like these phospholipid molecules. And they start to chip away at these phospholipid bilayers. So alcohols basically destroy the membrane and poke holes in the membrane. This is how 70% alcohol, if you use it on a bench top, or 70% alcohol on your skin, this is how it's killing bacteria. It's poking holes in those membranes, and now these cells can't control their own boundary. Detergents do the exact same thing. So you use soap and water, not just water, right? The cell wall that most cells have doesn't stop water from entering a cell. Water could enter the cell, but remember, the cell wall will give structure. So just cleaning your countertop with water ain't going to cut it, right? It's going to, these cells will still be fine. Most of them that have cell walls will not be destroyed. But if you have soap in there, a detergent, again, it has this kind of property that allows it to poke holes in membranes. Now, polymyxin molecules, these are actually derived from certain bacteria in the soil. Bacteria in the soil are competing for nutrients and space, and some bacteria in the soil make something like a polymyxin molecule. Now what we have done is we've borrowed that idea, because what these polymyxins do is they really attack gram-negative organisms. These polymyxin antibiotics, they take out the outer membrane, this membrane on the outside of the peptidoglycan layer. They chip away at the LPS molecules, those lipopolysaccharide molecules we talked about, okay? They attached on those, and they actually help with killing the cell, okay? So all of these are more related to the membrane than they are to anything else. Alcohols, detergents, and polymyxins all kill cells because they attack the membrane. Now, another prokaryotic envelope structure is an extension that is rooted in this envelope. So now in this image, again, I know, let's see, okay, again, I know that this is a gram-negative organism. The reason I know that is because I see a plasma membrane here on the inside, okay, and I see what they're saying is an outer membrane here on the outside, okay. So this has got to be a gram-negative bacterium. So one of the extensions that we see rooted in this prokaryotic envelope is a flagella, or I should say flagella or flagellum for singular. These provide motility in response to external stimuli. So bacteria can move towards things or away from things at will using these, basically they're like tails, right, that are built into the membrane and built into the envelope as a whole. So here we have a few examples of how these flagella are actually attached. We see these flagella can be attached just to one end. Okay, so if you look at trich or trike, here we have trichus at the end of every one of these words. Okay, and actually the root of this is sometimes used when talking about hair in anatomy and physiology. So it looks like a hair almost, right? These tails that are kind of hanging off. Mono in front of this word would indicate that there's only one. So that makes sense, right? There's only one flagellum. Amphi would indicate there's something going on both sides, okay? So we have our tails on both sides. Lopho is a bundle, okay? A bunch of them all together. So here we see, let's switch to red here for a second. So here we see that they're all kind of in a bundle at the end of the cell, in a bunch like that, all right? Actually, lopho means tuft or cluster, so makes sense. And then peri here, well, peri would indicate, let's use purple for peri, this would indicate that there's actually numerous spots around the cell that these flagella originate, okay? So lots of different ways these flagella can actually attach. And these provide for motility, for these cells to be able to move around. So what we do is we use terms like, we use terms with the suffix of taxis. Okay, so think of a taxi, right? So down here we see taxis at the end of these words. So think about taxis and they carry you around. So this is going to carry these cells around. If we think about a positive taxis, okay, some kind of movement in the direction of something, that would be positive, okay? If we'll switch to red for this, how about that, okay? If we have a negative taxis, then it's moving away from that thing, okay? So now we have to pick what is it moving towards or away from, okay? And this is where we run into this middle portion of the word, another prefix to the, so these would be prefixes to the word, right? So positive or negative would be placed in front. It's moving towards something or away from something. But then we could be talking about a chemical, about oxygen, light, temperature, and I'm sure there's more than just this list that I have here. And you'd put those as prefixes in front of taxis. So let's consider a chemical that might be harmful to a bacterium. Let's say it's bleach or something, right? Or an overabundance of a chemical that might be harmful. Well, this organism might want to move away from that. So we would say it's negative in that sense. And we'd say it's a chemical that it's moving away from, so that would be a negative chemotaxis, moving away from bleach. Let's say that a cell thrives in the presence of light, okay? So some bacteria, let's say, are photosynthetic, so they're like plants, right? They need sun to survive. So they might need to move towards the light. We would say that would be positive, right? It's moving towards something, and photo would be light. So we'd have a positive phototaxis. Right? So this is how we apply these terms, and we explain how it is that these are going to be modal organisms. You know, there's a motility towards or away from things. Now the last envelope extension, or a couple of different ones that I wanted to mention here. These extensions can allow for attachment. Okay, so one example would be Fimbriae. Now you might have heard of Fimbriae, maybe in anatomy you might remember that the end of the fallopian tubes had these finger-like projections that kind of shrouded that end of the fallopian tube around the ovary for the pickup of released egg. Okay, so here the Fimbriae are again kind of finger-like projections, but they look nothing like the end of a fallopian tube. Here we can see, I'll just use purple here to indicate all of these long extensions here are Fimbriae coming off of these particular cells. Now these are two cells. If I asked you what the shape was, what would you say? Hopefully bacillus, right? Now these two cells, you might think, oh, they're diplo, right? Diplobacilli, because there's two of them. But I think that these two cells probably are dividing. This is probably a process where one cell is giving rise to the other. So these would actually come apart here in the middle and become their own cells, okay? But these are Fimbriae that are all around this cell, and it helps for these organisms to attach to things. Again, this could be bad news for the host because this means that the organism can attach to the host and maybe infect the host, all right? But very important to that organism for survival. Now, another kind of extension that you could talk about are pili, or singular would be a pilus, okay? These are like a little cylinder that connect two cells to one another. So if we look here, we use green here. So way over here on the right side, you can see this kind of a hollow cylinder. All the way through here, you could have the movement of actual genetic material, okay? And it looks to me, as I look at this image on the right, that this organism on this top side seemed to have fimbriae. All right? And this particular cell is sharing something, possibly, with the cell that it has formed a pilus with. Okay? In this way, organisms share genetic material in the same generation. Okay? So sometimes I joke and I say pilus penis because the way that genetic material is transferred in humans involves the sexual activity, right? So you have the sperm and the egg ultimately coming together, right? And the donation of the male genetic material is done that way. Well, here we have a pilus that's formed between two cells, but the characteristics of the expression of those genes can be expressed in real time in this situation, right? In the case of sexual reproduction with the sperm, it's not until there's an offspring that the genetic material becomes apparent that it's having some effect. Here, it's possible that this cell above could share the genetic material that would allow this other cell to also make fimbriae and make it more virulent, make it able to attach to its host. So this is a really powerful kind of situation where two cells can join up and share genetic material, and that genetic material can go on to allow that cell to express those abilities in real time in that very same generation. Now, something before we finish up for this particular clip, something that's interesting and really medically relevant is that the ability to form fimbriae and pili like this are inherently something only pretty much gram-negative organisms do. We don't see fimbriae and pili formed by gram-positive organisms. All right? So this powerful ability to attach to the host, to attach to different things with fimbriae, this powerful ability to share genes in real time, they may be for the formation of fimbriae and attachment, but they could be for almost anything. These genes could give antibiotic resistance. I mean, the list will go on and on. We'll talk about it more later. But this is powerful, and this is more than anything a gram-negative kind of thing that goes on, not a gram-positive ability. Okay? So next time we'll pick up with some spores and we'll talk about how it is that gram-positives have something on the gram-negatives. Okay? We'll leave it at that for now. See you again later.